Hello everyone. Today we are going to discuss about post-fertilization structure and events. Okay, so let's discuss. First of all, we are going to discuss about endosperm. What is endosperm? Endosperm is basically a triploid structure that form during the double fertilization event in the angiospermic plants, and it help in the embryo development. Endosperm development precedes embryo development. That basically endosperm development that further help in the embryo development. And the primary endosperm cell divides repeatedly and form a diploid endosperm tissue जो हमारा primary endosperm cell है ना that will divide repeatedly to form a triploid tissue, and that tissue is filled with the reserve food material that is further used as a nutrition for the embryo. क्या है जो एंडोस्पर्म है वो डिवाइड करके क्या बनाएगा, प्राइमरी एंडोस्पर्म टिश्यू बनाएगा. द टिश्यू कंटेन फूड रिजर्व मटेरियल विच इज फर्दर हेल्प इन द embryo development. Okay, in the most common type of endosperm development, PN undergoes successive nuclear division. Then primary endosperm nucleus undergoes successive, means many, nuclear division to give rise to free nuclei. Basically क्या होगा भी, जैसे ये हमारा सेल है जो इसका एंडोस्पर्म है वो क्या करेगा, मैनी न्यूक्लाई डिवीज़न करके मैनी न्यूक्लाई बनाएगा और इसको हम कहते हैं फ्री न्यूक्लाई कंडीशन. फॉर एग्जांपल जैसे कोकोनट में होता है ना सिंशियम कंडीशन, फ्री न्यूक्लाई कंडीशन है ना, वो फॉर्मेशन होगा. ओके एक सेकेंड. This stage endosperm development is called as a free nuclear endosperm. और इसको क्या कहेंगे, फ्री न्यूक्लियर, क्योंकि न्यूक्लियर फ्री है साइटोप्लाज्म में, तो दैट इज कॉल्ड एज फ्री न्यूक्लियर एंडोस्पर्म. Sequentially cellular formation occur and the endosperm becomes cellular. Okay, so first of all, karyokinesis, that is a nuclear divides, and after free nuclear condition when the cell divides, basically cytokinesis occur that leads to formation of cellular endosperm. The number of free nuclei varies. The number of free nuclei formed before cellulation varies greatly. Yes, the coconut water from tender coconut that we are familiar with is nothing but free nuclear endosperm made up of thousand of nuclei. I have already said that endosperm of coconut is basically a free nuclear condition that is surrounding white kernel, that is called as white kernel is a cellular endosperm. That is very important. Now endosperm may be either completely consumed by the developing embryo. Basically endosperm is divided further, either it will be completely consumed during the embryo development or not. So for example, pea, groundnut, beans, in this endosperm is completely consumed for embryo development, okay, before seed maturation, or it may persist in the mature seed. Basically फॉर एग्जाम्पल कैस्टर, कोकोनट, एंडोस्पर्म प्रसिस्ट करता है, रिमेन रहेगा, इट विल नॉट कंज्यूम्ड लाइक पी ग्राउंडनट, इट विल परसिस्ट, विल रिमेन till the matured seed. For example, castor or coconut, as we also use the endosperm for eating purposes, right, in the coconut. So yes, and be used up during seed germination. That further endosperm will be used during seed germination. Split open some seed of castor seed, groundnut, coconut, look for endosperm. Yeah, that is okay. Now we come to embryo. First we have read endosperm. Okay, now embryo. Embryo develops at the micropyle end of the embryo sac where zygote is situated. So they have listed the location of the embryo. That embryo develop at the micropylar end of the embryo sac, the female gametophyte, where zygote is situated. Okay, most zygote divide only after certain amount of endosperm is formed. Basically zygote will start dividing only after endosperm is formed. This is some amount of endosperm is formed. This is an adaptation to provide a sure nutrition developing. Why zygote only divide after some amount of endosperm? For nutrition, because embryo needs nutrition, and endosperm is the only form that provide nutrition to embryo. That's why zygote only divide after endosperm formation, even if it is a little. Though the seeds differ greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledon and dicotyledon. So basically, they are saying that even both have different structure in monocotyledon, dicotyledon, one cotyledon, but still at the stage of embryo development, their embryogeny is similar. Their formation of embryo stages of embryogeny are similar in embryo development stage. Okay, फॉर एग्ज़ाम्पल इन्होंने इसमें दिखाया है 1.13 में, जैसे एम्ब्रोनिक स्टेज हैं वो सिमिलर हैं दोनों मोनोकोटिलिडन डाइकोट में. Now zygote will further give rise to pro-embryo that will further for globular, heart-shaped and mature embryo. Okay, let's see. For example, zygote, this is zygote, right? And zygote will divide by mitosis to form the embryo. So that is a two-cell stage, and further it will form a globular embryo. Okay, globular. We can also observe the shape of the embryo. Some look like a globular. That's a globular embryo. Now after that, heart-shaped embryo, because of similar to heart shape, and that is a mature embryo in which radical, cotyledon, palmules are visible. Okay, that is the embryonic stages of the after zygote division. Okay, now we are going to discuss about the dicot embryo. Dicot embryo consists of embryo axis that have a two cotyledon. For example, this is an embryo axis in which this palmule will form the shoot, and this radical will form the root, and it also contain a cotyledon. That two cotyledons are present. That's why it is a dicotyledon, presence of two cotyledon. Okay. Okay. The embryonic axis above level of cotyledon is called as epicotyle, which further form the palmule, or also called as stem tip, because palmule form क्या करेगा? shoot tip या stem tip form करेगा. The cylindrical portion below the level of cotyledon is hypocotyle. दिखाएं. For example, this is a one second. जैसे cotyledon है, the below the level of a cotyledon is a hypocotyle and above is a epicotyle, and for this is further forming palmule. Now below part further terminate into radical that form the root tip. The root tip also covered by a protective cap, root cap, to protect the ends of the root from any type of a destruction or any harm to the root. Okay, this is the structure of a dicotyledonous embryo. Now we come to monocotyledon embryo. Well, it possesses only one cotyledon. We have described it contain only one cotyledon. It belong to the grass family. Monocotyledons belong to the grass family that contain cotyledon, which is called as scutellum. What is scutellum? It is a cotyledon that is absorbed in the monocotyledon. Okay, where it is situated? It is situated on the one side, lateral side of the embryonic axis. As its lower end embryonic axis has the radical and a root cap that is further undifferentiated sheath called chlorhiza. Basically, for example, let's see, this is a monocotyledon embryo that belongs to grass, that belong to the grass family, right? So this scutellum that is present on the lateral side, one side scutellum, that is a cotyledon of monocotyledon. Okay, this is a coleoptile and this is a coleorhiza. What is coleoptile? Basically जो शूट अपेक्स है, ये शूट अपेक्स क्या है, पल्म्यूल है ना. ये coleoptile and coleorhiza, these are the protective layers. Coleoptile will protect the palmule. Coleoptile is a protective sheath that will going to protect the palmule, or coleorhiza is a protective sheath or layer that going to protect the radicle. So these are the protective sheath layer. And this is epiblast present at the in between of both, and this is a root cap protecting the tip of the root. Okay. The portion of embryonic axis above the level of attachment of scutellum is epicotyle. डेफिनेटली जो मतलब स्कूटैलम के जो अबव लेवल पे जो पोर्शन प्रेजेंट है उसको हम एपिकोटाइल कहेंगे. एपिकोटाइल हैज शूट अपेक्स एंड फ्ली लीफ प्राइमोर्डिया. व्हाट इज लीफ प्राइमोर्डिया? बेसिकली इट इज अ बिफोर द लीफ फॉर्मेशन इज अ लीफ लाइक pre-formation stage that going to form the leaf, precursor of leaf, we can say that. So yes, epicotyle contains shoot apex and the leaf primordia, which are enclosed in a hollow foliar structure called as coleoptile. क्या है coleoptile? क्या है? बेसिकली शीथ है, जैसे कोलोराइजा है रूट रेडिकल के लिए, वैसे ही पल्म्यूल के लिए या फिर एपिकोटाइल के लिए क्या है, कोलेप्टाइल है. Okay, now we come to seed. Seed, that is a character of angiosperms. It is a finally product of sexual reproduction, right? How seed form? That is formed during the sexual reproduction. It is often described as a fertilized ovule also, because it developed from the ovule. Fruit developed from the ovary, and seed developed from ovule. So yes, seed that are formed inside fruit are seed typically consist of seed coat. What are the layers? Seed coat, cotyledon and embryon axis. The cotyledon of embryo are simple structure, generally thick and swollen due to storage of food reserve. So seed contain seed coat, cotyledon, embryo axis. And what is cotyledon? That is a simple structure that contain reserve food material, right? For example, legumes, pulses, yeah, grams. These are, yes. Mature seed may be non-albuminous. Non-albuminous means they does not contain endosperm. Endosperm is completely used. Or ex-albuminous. Non-albinous seed have no residual endosperm. Basically, they does not have endosperm because it is completely utilized. It is completely consumed during embryo development. For example, pea, groundnut. And what is albinous? Where endosperm is present, it is not completely used during the embryo development. For example, wheat, maize, barley, castor. Occasionally, in some seeds such as black pepper and beet, remnant of nucellus are also persistent. That is also important. In which nucellus is persistent? That is a black pepper and beet. This persistent nucleus is the perisperm. Important. जो nucleus persist करता है वो क्या बनाएगा? Perisperm बनाएगा. Okay. What are integument? Integument of ovule that are hardened as a tough protective seed coat. Integument क्या बनाएगा? Seed की tough protective coat बनाएगा. The micropyle remain as a small pore in the seed coat. This facilitates the entry of oxygen, water into the seed during germination. जो हमारे एम्ब्रियो सैक होता है उस पर जो ओव्यूल होता है, ओव्यूल पे जो माइक्रोपाइल्ड होता है ना टिप रीजन पे जहाँ इंटेगमेंट एब्सेंट होती है, ओपनिंग होती है स्मॉल, तो वो क्या करता है सीड में स्मॉल पोरी बना के रखेगा. और वो हेल्प क्या करेगा? बट विल हेल्प, इट विल हेल्प इन द एंट्री ऑफ ऑक्सीजन, वाटर, बिकॉज इट हैज़ पोर, राइट. तो इट विल provide the entry of oxygen and water during the seed germination. As seed mature, its water content is reduced and seed become relatively dry. जब seed mature होगा, then water content will decrease and it become relatively dry. Okay. The general metabolic activity of embryo slow down. Oh, definitely, because it has become mature, right? Its metabolic activity will now start slowly down. It start decreasing. The embryo may enter a state of inactivity. What is dormancy? What is dormancy? When the embryo become inactive. Okay, or if favorable conditions are available, for example, moisture level is adequate, oxygen, suitable temperature, then they can germinate. Okay, what is dormancy? When the embryo become inactive due to unfavorable condition. If we provide the favorable condition, for example, favorable oxygen, temperature, humidity, moisture, then it will germinate. Okay. As a ovule mature into seed, the ovary develop into a fruit. That's the transformation of ovule into seed, ovary into fruit proceed simultaneously. The whole of ovary develop into the whole of fruit. Basically, the whole of ovary की जो whole है वो fruit की whole बन जाएंगी. For example, pericarp, right? The fruit may be fleshy, for example, in guava, orange, mango, or may be dry, for example, groundnut, mustard. Many fruits have evolved mechanism for dispersal of seed. Recall the classification of fruit and their dispersal mechanism that you have studied in the earlier class. Okay. ये फ्रूट फ्लैशी भी हो सकते हैं, फॉर एग्जांपल गुआवा, ऑरेंज, मैंगो, और ड्राई भी हो सकते हैं जैसे ग्राउंडनट और मस्टर्ड में. और इनमें डिफरेंट फ्लावर या फ्रूट या मतलब सीड का डिस्पर्सन मैकेनिज्म भी डिफरेंट है. सो ये हमने पहले पढ़ा हुआ है. सो नाउ डिस्कस द true or false fruit. Okay. In most plant, by the time fruit develop from an ovary, other chloroplasts degenerate, fall off, and the fruits will develop from ovary. Other parts, for example, sepal, petal, they have they will basically become mature old. They will fall off, right? However, in a few species such as apple, strawberry, cashew, the thalamus also contributes to fruit formation that is called as false fruit. When fruit form from an ovary, listen, what are true fruits? When the fruits form only from the ovary. Fruits that form only from the ovary, that is called as true fruits. What are false fruits? When the fruits form any part other than ovary, then it is called as false fruit. For example, it can also form from thalamus in such as apple, strawberry, cashew. In this fruit form from thalamus. Okay, so that's why it is called as false fruit, because fruit is not forming from ovary in apple, strawberry, and cashew. Although in most species fruits are a result of fertilization, definitely fruits form as a result of fertilization. There are few species. We can also see here, this is the thalamus part. For example, this in the apple, and this is the strawberry, that is also eatable, right? Thalamus part. In this fruit also thalamus part. Yes. In which, what are parthenocarpic fruit? The fruit develop without fertilization. Fruit is developing without fertilization, that is called as parthenocarpic fruit. And in this type of fruit, seeds are also not formed. They are seedless. For example, banana. Parthenocarpic can be induced through application of growth hormone. Basically, parthenocarpic, the fruit formation without fertilization, that can be induced by the external application of growth hormone. For example, gibberellin, right? That is also a growth hormone. And such fruits are seedless, definitely, because no fertilization has takes place. So how seed can form? So that's why they are seedless. Seeds are often advantage to angiosperm. Firstly, their reproductive processes such as pollination, fertilization are independent of water. Seed formation is more dependable. Also, seeds have better adaptive strategies for dispersal to new habitat and help the species to colonize another area. They have sufficient food reserve. Young seedlings are nourished until they are capable of photosynthesis on their own. Basically, seeds are offer advantages to angiosperm. They act as a reproductive process, for example, in pollination and fertilization. But seed formation are more dependable, right? That's why they have to adapt many adaptive strategies for the dispersal of to new habitat. But they seed in itself contain food reserve so that young seedlings, for example sprout, we can see in the in the moon when we grow they develop into beautiful sprouts. We definitely see in it many times. So they also contain food reserve for the young seedling that are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to young embryo. Seed coat क्या है? That help in the protection of the embryo. Being product of sexual reproduction, they generate new genetic combination leading to variation. Seed that is result of sexual reproduction, and we know sexual reproduction help in the variation. So yeah, that is definitely advantages. Seed that is the basis of our definitely agriculture. Dehydration, removal of water, and dormancy, inactive state of embryo of the mature seeds are crucial for storage of seed. We have basically these are the techniques to store the seeds. For example, a seed is inactive in the dormancy, we can conserve or preserve that or store that, right? Or when they dehydrate or become dry, right? We can store it, which can be used as food throughout the year. We many different type of seeds, so that can be used as a food purposes or also to raise crop in the next season. Definitely we can use for next season. Okay, so that's all for today. Thank you so much.